Protein phosphorylation refers to the process of transferring ATP phosphoric acid group to amino acid residues serine, threonine, and tyrosine in the base protein catalyzed by protein kinase, or binding GTP under signal action. Protein phosphorylation is a common regulatory mode in organisms and plays an important role in the process of cell signal transduction. Protein phosphorylation is the most basic, common and important mechanism to regulate and control protein activity and function. Protein phosphorylation is the most widely used type of post-translational modification in signal transduction. Protein phosphorylation affects every basic cellular process, including metabolism, growth, division, differentiation, movement, organelle transport, membrane transport, muscle contraction, immunity, learning, and memory. The figure shows the basic catalytic cycle for substrate phosphorylation by a kinase. Starting top left, ATP binds to the active site of the kinase. This is followed by binding of the substrate to the active site. Once bound, the gamma phosphate of ATP, which is red ball in this figure, is transferred to a serine, threonine, or tyrosine residue of the substrate. After phosphorylation, the substrate is released from the kinase. The last step shown is the release of ADP from the active site. All protein kinases have a similar protein fold that comprises two lobes. One lobe shown blue consists of mainly beta sheet structure, and the other lobe shown green, orange, and yellow consists of alpha helices. This lobe structure forms an ATP binding cleft that constitutes the active site. The crystal structure of CDK2, which is short for cyclin-dependent kinase 2, shows this representative fold. The red ball and stick modeled bound in the cleft represents ATP. Despite sharing a common fold, kinases bind to and phosphorylate different protein substrates, partly due to differences in the charge and hydrophobicity of surface residues. Electrostatic surface representations of 4 different kinases are shown. Blue indicates positively charged surfaces, and red indicates negatively charged surfaces. The 4 kinases in the picture represent 3 serine/threonine kinases CDK2, pKa, PHK, and 1 threonine protein kinase IRK. Protein phosphorylation alters local charge interactions that are important in establishing specificity. The figure shows the ribbon diagrams, electrostatic surface representations and schematic diagrams of the substrate binding site of CDK2 bound to a substrate peptide. The positively charged lysine in the P+3 position of the substrate interacts strongly with the negatively charged phosphate on phosphothreonine 160 of CDK2. If CDK2 is not phosphorylated at threonine 160, it has a much lower binding affinity for peptide substrates and shows no preference for a positively charged amino acid in the P+3 position.